So good evening all, and I welcome you to Agri-Addict Plant Science online classes. So with the ongoing discussion today, we will discuss a little topic about population genetics, where we'll be covering the Hardy-Weinberg law, right? So before directly jumping into the Hardy-Weinberg law, as I told you that it deals with the population genetics, right? So whereas in case of your crop improvement, you will be in case of self-pollinated crops, you will be focusing on a single plant and then forwarding it to the next generation and then so on, right? But in case of population genetics, the name itself indicates that you are focusing on the entire population, right? Your focus will be on total population. So what is this population, and Hardy-Weinberg law applies to which population, right? So this is the one we need to know at first. So can anyone tell what do you mean by random mating population? Anyone, whatever it might be.
Sir, all possible crosses.
Okay, all possible crosses. Still any answer? Like someone told it is all possible crosses. So what do you mean by random mating? First, let us discuss about what do you mean by random mating. So the student who just said that it is all possible crosses, yes, it is right. But in order to put it in an effective manner, we can say that in case of this random mating, what happens now? So every individual, right, it has the equal opportunity to mate with the other individuals in the population, right? This is what you call random mating population. Okay, so this Hardy-Weinberg law applies only in case of random mating population. And then this random mating population, so where we can easily define that in a population, in a group of population, every individual has equal chance to mate with the other individuals, right? So this is what you call it as random mating population. This random mating population, it is also known as pan-mictic population or Mendelian population. Okay, so the Hardy-Weinberg law applies only in case of random mating population, and this random mating population, it is also known as pan-mictic population and Mendelian population, right? So please also note that the term pan-mictic, it was given by a scientist named Weismann. Okay, and then the term population, it was given by Johansson. In case of this Hardy-Weinberg equilibrium, right? Pan-mictic by Weismann. So Hardy-Weinberg law applies only to random mating population, which is also known as pan-mictic or Mendelian population. So can we also call the population as the gene pool? Anyone? Any idea? So can we call the population as gene pool? See, we can call the population as gene pool when we have the individuals of same species and they reside in the same gene pool or they share the same gene pool. Then individuals which have same species and share the same gene pool can also call this as the population. Okay, so please to remember this, where the individuals are of same species and share the same gene pool, right? So this is just the population. And then our Hardy-Weinberg law, it applies only to the random mating population or pan-mictic population or Mendelian population, right? So we'll enter into the different terminologies that are being used in this Hardy-Weinberg law. So there are two terminologies. One is gene frequency and second is genotype frequency. So can anyone say what do you mean by gene frequency in simple language? Can anyone describe it? What do you mean by gene frequency? Guys, am I audible?
Yeah, sir.
Okay. So can anyone tell me what do you mean by gene frequency in simple terms? Okay.
The allelic frequency,
Yeah, it is also known as allelic frequency, but how do you define?
proportion of that alleles in the population.
Okay. Yeah. So proportion of alleles of a gene, right? In a which population? In a random mating population. Okay. Because we are discussing about the Hardy-Weinberg law. So let us consider it is a random mating population, right? So the proportion of alleles of a gene in a random mating population. This is known as gene frequency, and this gene frequency, it is also known as allele frequency or allelic frequency, right? Some people also call it as genetic frequency, but most used term is your gene frequency, or it is also known as your allelic frequency. All right. Okay. So how will you calculate this gene frequency? So let us take an example. For example, in a population of 100 individuals, right? So let us consider that red color is dominant and white is recessive. So the red, and so we'll go with the homozygous thing first. And also consider this, there are heterozygotic individuals in this 100, right? So if there are 30 homozygous red individuals and then 30 homozygous white individuals, and remaining will be 40 heterozygous individuals, right? So what is the allelic frequency of capital R? That is your dominant allele. How will you calculate it?
Sir, 0.5.
Okay, how?
Sir, considering total number of capital R and half of the capital R smaller by total population.
Okay, so 30 plus you told 20, half of this, right? By total population. So that will be your 0.5. So this is one technique to calculate, and then other technique is 2 into, so the homozygous dominant individuals, it will be 30 plus, we will calculate the entire heterozygous individuals, because even in this case our capital R, that is the dominant allele, is being included. So 40 divided by 2 into total number of individuals. Okay. So eventually we will end up with this value. So this is how you will calculate the gene frequency or allelic frequency, right? Hope you guys got up to this. So how do you define then the genotype frequency? So one told that the proportion of alleles of a gene in a random mating population is your gene frequency. Okay. So how would you define genotype frequency? Any idea? It is the same thing. Instead in the place of your alleles, you will just substitute the genotypes, right? So the proportion of genotypes of a random mating population. So this will give you the genotype frequency. Is it okay?
Yes, sir.
Right? Okay. So in case of genotype frequency, it is also known as zygotic frequency. Okay. Genotype frequency, it is also known as zygotic frequency, whereas in case of your gene frequency, it is also known as allelic frequency, because the gene, it has two alleles, right? But the genotype forms only when two alleles come together, right? That is, whenever the zygote develops into a individual, then we can calculate the genotype frequency. So proportion of the genotypes in a random mating population gives the genotype frequency, which is also known as your zygotic frequency. So whereas in case of this example, the same example, so what is the genotype frequency of heterozygous plants? Anyone? What is the genotypic frequency of heterozygous plants?
0.4.
Okay, so that will be 0.4. So in the same way, the red as well as your white will be 0.3 and 0.3, right? So these are two different terminologies that we need to know before entering into the Hardy-Weinberg law. So now coming to the Hardy-Weinberg law. So I think in 2021 or 22 paper, they just asked about the scientist who gave this law, right? So the name of the law itself indicates the name of the scientists. One is Hardy and the other was Weinberg, right? So Hardy, he was a British mathematician. Please note that. I think this question was asked. Hardy was the British mathematician, right? Whereas Weinberg, he was a German physician, right? So Hardy was the British mathematician, and then Weinberg was a German physician. So both discovered the law. He discovered it in England and he discovered it in Germany independently, right? Do anyone know the year when they discovered this law? So the year of discovery was 1908. Okay, and they together commonly named this principle as Hardy-Weinberg law. So please to remember, Hardy was the British mathematician and Weinberg was German physician, and the year of discovery was 1908. So someone tell me, what does this law states? What does this law state? Or what is the actual meaning of this law? Anyone? Okay, someone wrote 1908, right? So how do you define Hardy-Weinberg law? You see, this is the important understanding. How would you define Hardy-Weinberg law? Anyone? See, Hardy-Weinberg law defines that in a large random mating population. Okay, so as we discussed in the initial slides, the population should be in random mating. So in a large random mating population, and the terminologies that we discussed was gene frequency and genotype frequency, right? So in a large random mating population, the gene frequency and the genotype frequency should remain constant or it should not change, right? Generation after generation. Okay. So this is the basic law. So in a large random mating population, the gene and genotype frequency should remain constant generation after generation, right? If there are no selection, mutation, migration, and genetic drift. Please do remember these four. Okay, so these are most important things. So in the absence of this, then only this law works, right? Our Hardy-Weinberg law, it states that in a large random mating population, right, our gene and genotype frequencies, they remain constant, right? They remain constant generation after generation. Okay, so they will remain constant generation after generation when there is no selection, no mutation, no migration, and no genetic drift, right? So you should not apply any selection, and there should be no mutation in the population, there should be no migration, and there should be no genetic or random drift in your population, right? So this is the law. You need to remember every single word, right? In a large random mating population, our gene and genotype frequencies, they remain constant or unchanged generation after generation if there is no selection, mutation, migration, and genetic drift, right? Is it okay? Did you guys understand what Hardy-Weinberg law defines? Just, is it okay, guys? Hello? Okay. So this is
Yes,
what
sir.
your, yeah, okay. So this is what the law states or defines, okay? So in a large random mating population, both our gene and genotype frequencies, they remain constant generation after generation if there is no these four forces involved, okay? So these forces should not be acting on our random mating population. Then our gene and genotype frequency, it remains constant generation after generation, right? So please remember these four forces. Okay, so selection, mutation, migration, and genetic drift. So now let us get into an example. So let us assume that these are the parents. So the homozygous dominant and then the homozygous recessive. So parent 1, parent 2. When we cross, so what do you call this generation? If we cross between two parents, the obtained generation, what do you call this? F1, right? So this is your F1 generation. And when you self this F1 generation, or if you leave the F1 generation for random mating, whatever it might be, okay, if you self or if you leave the F1 generation for random mating, so you will be actually ended up with four different genotypes in which generation? In the F2 generation, right? So this will be your parental generation, F1 generation, and then your F2 generation. So let us assume that the frequency of the dominant allele, please be attentive here, the frequency of the dominant allele, okay? So the dominant allele will be capital A. So the frequency of the dominant allele, for example, let us assume it is B. Then what will be the frequency of the parent one Means what will be the genotypic frequency of parent 1?
P square.
Okay. So which is nothing but combination of two alleles, that is your P square. So in the same case, if you consider that the genotype small a, and then the allelic frequency of small a is Q, your genotypic frequency of parent 2 will be Q square. So with this understanding, what would be the genotypic frequency of your F1 generation? It will be PQ, right? And then coming to
Yeah,
the
sir.
F2 generation, we will be seeing again P square. Here it will be PQ, and again here it will be PQ. Here it will be Q square, right? So they said that it is a mathematical relation to describe this Hardy-Weinberg equilibrium, that is between these alleles. So that is a mathematical relationship, okay, to describe the equilibrium between these alleles. So here in this example, we have just considered a single gene, right? At a locus it has capital allele, and at the other locus it has the small allele, right? So a single gene with two alleles, that is your capital A and then small a, just for our assumption. And we also assumed that this will be the gene frequency, and this would be the genotype frequency. Same applies here. And so what does this indicate? Whether it indicates a gene frequency or genotype frequency? PQ.
Genotype.
Right. So it indicates your genotype, or which is also known as your zygotic frequencies. Right? So the mathematical relation that they described is always the gene frequencies when combined will be equal to 1. Point number 1. Okay. And even in case of this panmictic population or random mating population, right, all the genotypic frequencies when summed up will also be equal to 1, right? So this can also be derived from this, just like what will this expression get modified when we compress it? It will be P plus Q whole square, that will be 1. So eventually P plus Q will be equal to 1, right? Okay. So just pay a little more attention here. I'll tell you a thing. So here the genotype frequency in case of the F2 generation, the genotype frequency of homozygous dominant will be, what will be the genotype frequency?
P square.
Okay. And what will be the genotype frequency of the homozygous recessive?
Q square.
So what happens with the genotype frequency of your heterozygous in the F2 generation?
2PQ.
Okay, right? So this is the thing that you need to notice. In case of the random mating population, the genotypic frequency of your heterozygous individuals will not be PQ, it will be 2PQ, right? So we have two different, means the same genetic frequency, but it is 2 times, right? So the genotypic frequency of our homozygous dominant will be P square, followed by the heterozygous individual will be 2PQ. and then the homozygous recessive will be Q square. So everything when summed up will be equal to 1, right? So P square plus 2PQ plus Q square is equals to 1, and then P plus Q is equals to 1. Okay, so even if you allow this F2 generation for random mating, okay, and then if you calculate what is the different genotypic frequency of the different individuals, you should end up with this relation, because if the population is undergoing the random mating, the gene as well as the genotype frequency will not change. Okay, so the gene frequency as well as your genotype frequency, so on. Okay, until you allow the population to continue it in the random mating, you will see no change in the gene and genotype frequency, right? But whenever the evolutionary forces such as selection, mutation, migration, and random drift or genetic drift, any of these four forces act on your population, then your population from next generation will not be in the Hardy-Weinberg equilibrium. Is it okay? When this selection, that is your evolutionary forces act, the population will deviate from the Hardy-Weinberg equilibrium, right? So if the population gets deviated from the Hardy-Weinberg equilibrium, how would you achieve the next generation to be in Hardy-Weinberg equilibrium, or what would you do to maintain the population in the Hardy-Weinberg equilibrium? Anyone? Let us consider that your population, in your population there is one mutation, or someone selected a plant and forwarded it, whatever it might be. The four evolutionary forces, among these four evolutionary forces, one force has changed the Hardy-Weinberg equilibrium of your generation. Then how would you ensure that the next generation will again come to Hardy-Weinberg equilibrium? Anyone? Any idea? So if your population is acted by any of these four evolutionary forces, just if you want the next generation or the subsequent generations to be in the Hardy-Weinberg equilibrium, you just allow all the plants to random mate. Okay, random mating is the principal component in this population genetics to achieve the Hardy-Weinberg equilibrium, right? So just leave the plants in random mating, you will eventually achieve your population in the Hardy-Weinberg equilibrium, right? So this is the thing, and we'll just go through a simple calculation. So with the discussed matter, I hope you will be able to calculate the frequencies of the other two genotypes. Just go through this problem and let me know what would be the results. Someone just wrote 0.64. So explain me how, what is 0.64? Just tell me. How would you calculate? How would you conclude 0.64? 0.16, someone told 0.16. Okay, so you guys please explain, or you can also either typing it in the chat box. So explain in the chat box. 0.48 heterozygotes, right? Now explain me how you got these 0.16 and 0.48. And someone told 0.64. How would you conclude that it is 0.64? Okay. So it has clearly mentioned that a population of trees has two alleles for a particular gene that controls the seed cover. So the dominant gene results in the green seeds, while the recessive results in the yellow seeds. If the frequency of homozygous dominant genotype, which is nothing but your GG, is 0.63. So which means, so if you convert it into the genotypic frequency, let us consider that the genotypic frequency of this will be p square. So p square is 0.36. So your p will be 0.6. So according to the Hardy-Weinberg law, so p plus q will be 1. So if p is 0.6, then q will be 0.4. Right? So once you are obtained with the allelic frequencies, let us proceed to the genotypic frequencies. Right? So he told that this is one genotypic frequency, as he mentioned, it is 0.36. So the other remaining two genotypes will be this and then this. So now tell me, what will be the genotypic frequency of heterozygous individuals if this population is in random mating? I just want the expression, right? What will be the genotypic frequency of your heterozygous individuals if the population is in random mating? No, it will not be PQ. See, it will be 2PQ. Please have this idea as we discussed even here. So the heterozygotic individuals, the genotypic frequency will be 2PQ. Okay. So if we go with the 2PQ formula, we will be obtaining 2 into 0.6 into 0.4. Right. So you will end up with 0.48. Right. So whereas in case of this homozygous recessive genotype, it will be your Q square, that is equal to 0.16, right? So the three genotypic frequencies will be this, right? Okay. So this is your questions from this chapter will not be much hard. This will be in the minimal range, but just remember, in case of the heterozygotic individual, your genotypic frequency will be 2PQ, right? Okay. Um, so I hope now you are able to calculate the questions from Hardy-Weinberg law. So I'll just try to give you another question. So calculate it. So just see this. In a population of 100 individuals, okay, so there are 50 homozygous dominant. Okay, in a population of 100 individuals, there are 50 homozygous dominant individuals, right? And there are no heterozygotes. Right, so you can easily tell what will be the individual number of homozygous recessive. It will be 50, right? So in this case, what is the gene frequency of dominant allele? How would you calculate the gene frequency of dominant allele? Okay, 0.5. All right. So if I say a statement that 0.5 is considered as the maximum gene frequency, right? Is this statement correct? Not only in this example, but it is a general statement that 0.5 is considered as a maximum genotype frequency in an Hardy-Weinberg equilibrium. Will this statement stand true? Okay, no. Why? Why it can't? Okay, if no, so what would be the maximum gene frequency that we can see in Hardy-Weinberg equilibrium? Because it can up to 0.7. Okay. Okay. If it is not true, so what would be the maximum gene frequency or allele frequency that you can see in Hardy-Weinberg equilibrium? Anyone? 0.7. Okay. So why only 0.7? Can't it be 0.8 or 0.9? See, in case of Hardy-Weinberg equilibrium, the population, it should be undergoing random mating. So the gene frequencies will constantly remain unchanged generation after generation. So with this law, you can easily say that the gene frequency maximum can go up to any number except 1, because if one allele is completely fixed in the population, okay, so there will be no random mating. Except 1, it can take any number. Okay, so just try to solve the other question. So in the same question, if I replace the genotypic frequency of these homozygous dominant genotype with 0.25, then what would be the genotypic frequency of other two genotypes? If I just replace the 0.36 with 0.25, then what would happen? Tell me the genotypic frequency of other two genotypes. Okay, someone wrote 0.25 and 0. Yeah. So 0.25 recessive and 0.5 heterozygotes, right? Okay, just a minute. So let us consider the same example, and if now there is no genotype frequency given, but the only thing that we got is gene frequency. If the gene frequency of the dominant allele is 0.9, okay, so what would be the genotypic frequencies then? What would be the three genotypic frequencies if the gene frequency of your dominant allele is 0.9? If your gene frequency is 0.9 for the dominant allele, so what would be the genotypic frequency of the three different genotypes? Zero point 0.81, 0.18, and 0.01. Okay. So tell me in terms of the individuals then. In terms of individuals, means how many will be the homozygous dominant, how many will be heterozygous, and how many will be homozygous recessive. So now you just indicated in the terms of frequency. In individual perspective, how many it will be? So it will be 81 individuals for your homozygous dominant and 18 individuals for your heterozygous and only one single individual for the homozygous recessive, right? So mostly your problem will be only in this range. So please do not miss any question if the problem comes. The only thing that you need to remember in this is, so the heterozygotic individual gene frequency, the genotypic frequency will be 2pq, and then the four different evolutionary forces. To remember all these four, followed by the name of the scientist and the country, right? And then the year in which they commonly described this principle. Okay, right? So this will just cover your Hardy-Weinberg law. So if you guys have any doubts in Hardy-Weinberg law, you can just ask me. If you find any difficulty later, you can text me in the WhatsApp group. Okay. So I saw someone was asking about parameterization. Can anyone answer what do you mean by parameterization? How will you describe parameterization? Anyone? If not, guys, please do refer it in the book, or you can also search in the internet. It is just a single statement. With that, just a single statement, you can know what do you mean by parameterization. Okay, and it is also considered as an exception to Mendel's first law. Okay, the only exception to Mendel's first law is the parameterization. So what would be the exceptions to Mendel's second law? What is Mendel's second law? A mutation which is caused by other alleles in same cell. Okay, you can just go a little bit deep, then you can have a clear understanding. You have a superficial understanding. It is correct mutation which is caused by other alleles in the same cell. Okay. So this is the only paramutation is the only exception to Mendel's first law. So what is Mendel's second law known as? What do you call Mendel's second law? Law of segregation, the Mendel's second law. This is the second law known as law of segregation. Okay. So the Mendel's second law is your law of independent assortment, right? So tell me one exception to this law of independent assortment. Linkage, right? Okay, so the main exception to this independent assortment is the linkage itself. Okay. So if anyone asks you what are the linkage group in case of Arabidopsis, what will be your answer? Linkage group in Arabidopsis. You need to know that Arabidopsis is a model plant in plant kingdom, right? So it will be 4. So linkage group will always be equal to your haploid chromosome number. So in case of this, the Arabidopsis haploid chromosome number will be 4. All right. Then what is the linkage group in case of Pisum sativum? Yes, 7. All right. So this you need to know, right? And so if I say statement that uh, maternal effect is governed by nuclear genes, is the statement right? No. Okay. So if I say a statement that the cytoplasmic male sterility is due to mitochondrial and chloroplast genes, is that statement right? Cytoplasmic male sterility is due to mitochondrial and chloroplast genes. Okay. The mitochondria. So can you name a membranous organelle in cell? Which organelle does not have any membrane? Centriole, and then allow the nucleolus ribosome. Okay. So just wait a minute. I just received a question from someone stating. Okay. So listen to the question carefully. Futures of cell theory. Okay. So the futures of cell theory are. I'll just write. Okay. Features of cell theory are: point number 1, plants and animal cells differ superficially but have same level of organization. 2, both plant and animals are made up of cells. And point number 3, so the cells here can assimilate, they can respire and respond to change in external environment. So the question is this: among this, how many statements are true and how many are false? Anyone? So let's get to the question. We also gave you all three correct. Okay. A, B, true. Alright. Okay. So according to someone, they told that all the three are right. And are right. And someone told that A and B are true. So A, B, true. Alright. Any still any deviation from all three correct and A, B, true? Is there any deviation? Am I audible now? Okay. So tell me, what are the three points of cell theory? What are the 3 points of cell theory? Or you can just write down. Cells arise from pre-existing cells. Alright. Next. Only that both plants and animals made up of cells. Alright. And the other? Structural and functional unit of living organisms. Okay. So now we'll just go to the question where it has given that the following are the features of your cell theory. Point number 1, plant and animal cells differ superficially but have same level of organization. B, both plant and animal are made up of cells. And C, cells can assimilate, respire and respond to change in external environment. So among these three, how many will fall under the cell theory? Do you guys think all three will fall? Hello? First two. So how first will come under the cell theory? Where it is given that the plant and animal cells differ superficially but have same level of organization. It was not proposed in the cell theory, right? The first statement of your cell theory states structural and functional unit of living organisms are these cells. So the question that I wrote, only the statement 2 satisfies your cell theory. Okay. So someone also said that 3. So how 3 will satisfy? See, the cells can assimilate, respire, and respond to external change. Okay. So I don't think that it was proposed in the cell theory. In case of cell theory, it was just given that both the plants and animals are made up of cells. Cells arise from the pre-existing cells, and structural and functional unit of the living organisms are your cells. That's it. Okay. So there is no deviation. Only 2 will be correct in case of cell theory. So if I just remove the futures of cell theory, then how many statements will be correct? All right. All. Okay. So if you carefully see the third statement, so in case of the first 2 statements, it is clearly written that plant and animal cells differ superficially but have same level of organization. So this is true. So superficially means from the outer layer. In case of plants, we will be having a cell wall, whereas in case of animal cells, there will be no cell wall, right? Whereas both plants and animals are made up of the cells. So when coming to the third statement, cells, where it indicates both plant and animal cells can assimilate. So the meaning of assimilation here is to produce food, okay, to maintain by itself. So can the animal cell perform or can produce a food by itself? Okay, so you need to be careful with this type of questions. First, read the heading that is being given. Okay, in case of which of the following features of cell theory are true. So these are just to confuse you. Okay. You also received some other questions. Let me tell you. Dore. Okay, most of the questions are answered by others. It means so I think there are no questions remaining. So with this, I'll be concluding today's class. Okay, so I'll be concluding today's class. Thank you all for attending.